Today's topic is cell communication. This sort of wraps up our series of videos on cellular processes that we've been working through, looking at biochemical processes within cells and how they work. So this is kind of the follow-up to respiration and photosynthesis. The good news is this is not as difficult as photosynthesis and respiration to understand. You already have a lot of chemistry background knowledge to help you. Before we begin, remember all cell signals are chemicals, and I think from baby bio on you're used to the idea that cells can send signals to one another in the form of chemicals, and that when those chemicals are received, they force some sort of action within the cell. So to begin with, one easy way for chemical signals to be passed short distances via paracrine signaling, right? Remember that from our POGL the other day, is using the cell junctions, right? Remember back from our membranes unit, we talked about the different kinds of junctions, the ways that cells are linked together. Specifically, what I want to focus on is the plasmodesmata, right? The plasmodesmata of plant cells, and the gap junction of animal cells. Okay, these two junctions are shown in the image below our slide here. The plasmodesmata, remember, are holes that exist. They cut across not only the membranes but the cell walls of plant cells themselves. This is an electron microgram of what the plasmodesmata look like, and this is what they look like in an artist rendering. So this is between two plant cells, and essentially notice that the cytoplasm can flow freely through the plasmodesmata, as can other cellular parts. The smooth ER of one cell can actually kind of go on through via these tubules to directly communicate with the smooth ER of the next cell. In animals, a similar type of junction exists, only this one is called the gap junction. All right, and here again you've got an electron micrograph of what that looks like, and then this is the artist rendering. Instead of having a wall, you don't have a cell wall, so there's that little gap between the cells here, but that gap is bridged by a protein rosette that you see here, and this is what it looks like from one side, but then when you cut it away, you can see that it's essentially a channel through which cytoplasm, chemical signals, whatever, can flow. So you can do short distance signaling in that manner. All right, now cell communication itself, as you saw from the TED Talk on bacterial communication, is believed to have evolved in prokaryotes and then possibly even some single-celled eukaryotes. You can have differences in the distance through which signals are sent. You can even have cells signaling themselves in the case of autocrine signaling. You can have it be kind of short distance, real close by local signaling that we call paracrine. And then you can have long distance signaling where you have, say, a brain or controlling the release of hormones from a gland, and then the target of those hormones is in another part of the body, and that we call endocrine signaling. Okay, so we've got different distances across which we can signal. So that's what we mean when we say paracrine. You also have some synaptic signaling. Recall that the junction between two nerves is called a synapse. It's really a gap between two nerve cells, and the axon of one nerve will send neurotransmitter substances across that synapse. They'll release substances into the synapse, and those substances get picked up by the dendrite of the next cell. Hormonal signaling that I mentioned before, that's called endocrine signaling. All right. And just as a quick recap, single-celled organisms do use communication in quorum sensing. So bacteria can sense how many of the same species are present and determine if they have enough power, basically, to overcome a person's immune system and turn virulent, right? And you saw that in the video. Sometimes bacteria have to communicate to coordinate activities if they need to. You have some organisms, some bacteria will live in what we call a biofilm, and they can live on the surfaces of your teeth. They can live on the surfaces of water and places like that. And really, in a biofilm, the bacteria act kind of like one organism. It's like a filmy colony of bacterial cells that will communicate with one another and divide labor and so on so that they can function more efficiently. And you had a whole big master class on that in the TED Talk that you watched. All right, so let's talk now about signaling in multicellular organisms. There are three basic stages, right? You receive a signal, that's reception. So usually that's done by a protein on a cell surface. Sometimes it's actually the receptor protein that is supposed to receive the signal might be inside of the cell, and we'll get into that in a minute. Transduction occurs where that surface binding causes a change in the configuration of the protein receptor, which initiates kind of a biochemical pathway, if you will, and that's kind of what you're seeing here. So here's the signal, and remember we call the signal a ligand, Ligand, right? So a cell signal, we call that a ligand. And this you got from your POGL that you did on, I guess today you did that POGL. All right, so the signal is received here, that's reception, right? The signal then, once you have this conformational change, it gets transduced, or you start this biochemical pathway. And then often that signal can be amplified, so notice one molecule produces 4, each of those produce 2 more, and so on, until you get to the point where you have a buildup of a certain molecule inside of the cell, and in response, the cell will do something that was what we originally wanted it to do. Okay, so you get a cell's response. So, for example, if you drink milk, lactose gets received. Lactose becomes the ligand. It's received by a receptor, and that's going to trigger a series of chemical reactions that once you build up the last step in that chemical reaction, that's going to trigger the cell to start producing the lactase enzyme so that all of this lactose that you've just introduced to your body can be broken down. All right, now I mentioned some receptors are on the surface of cells, and then some of them are inside of cells. Okay, so here is the term ligand that we were talking about. This is the signal molecule, and the ligand typically, here's an example here, will bind to a receptor. Now this particular receptor is known as a G protein. There are not all G proteins, but G proteins, that's what this is, act as a switch to turn on or off the internal portion of the receptor. So when the ligand binds into this receptor, what it does is forces this G protein to let go. Okay, and when it does, that lets go and will bind to another enzyme, which triggers then transduction. Okay, so that's a more complicated example. It doesn't always work that way, but if you understand that, it's really easy to understand how a ligand binding to a regular receptor could cause a conformational change like you saw in the previous picture that we looked at. Now, when we're looking at steroid hormones, these are the lipid-soluble hormones, their receptors are actually inside of the cell because remember, steroids are small and nonpolar, and therefore they can easily, here's an example of it, the steroid hormone can pass through the membrane quite easily. This receptor protein, instead of being bound to a membrane, is actually just floating in the cytoplasm. When it binds to a hormone, that becomes an activator that will target a specific gene in the DNA molecule and force it to begin making copies of the RNA that we need to create whatever protein that is signaled to be produced by this particular steroid hormone. Okay, so again, this ligand is a steroid, and that can go inside and be received, whereas protein hormones that are made of proteins, they have to bind to a surface receptor. All right, so now we get to the meat of the matter, the signal transduction pathway itself. Okay, so here we've got a ligand, it's bound to this receptor. And what this does is activates some molecule here. Now, what you saw in your models, that activation takes an inactive protein and phosphorylates it. So we take an ATP molecule here and strip it of a phosphate, releasing it as ADP. But now that phosphate is bound into this protein, which activates it. That then will catalyze the reaction of this inactive protein to be phosphorylated, and so on until you get to the end where you get a response triggered. So most of the time what you end up with are what we call a series of protein kinases. This protein kinase transfers phosphate groups from ATP to protein, which would activate, this then becomes the protein kinase for the next inactive protein. This becomes the protein kinase for the next inactive protein, and so on down the line. Okay, and that's why we say this is a biochemical pathway, because it involves a number of these kinds of reactions. Sometimes what you have is called a second messenger system, where instead of something happening immediately right here, what you end up with is a small ion produced, or something or you'll actually create an enzyme that produces a small ion or some sort of other molecule, like C-AMP, cyclic AMP, which would trigger the protein kinases to start working and to get a signal transduction pathway. So instead of the ligand binding being the thing that starts off this signal transduction pathway, the ligand binding produces a second messenger instead, and it's that second messenger that would start our phosphorylation cascade. All right, so responses by the cell are triggered either in the nucleus or on the surface of the cell, depending on what the ligand is, if the ligand's capable of getting through. Now, often the cell has to amplify the response. And that's what you saw way back in the first picture I mentioned, where you signal transduction involves the production of one molecule, but then that molecule produces 2 or 4 other molecules, and each of those produces 2 or 4. So what you're doing is each step in the process, you're increasing the concentration of the activated enzymes, which produces even a stronger signal in the cell. There's also such a thing as specificity. So certain proteins have a more specific effect depending on which cells they're received in. So epinephrine, has one effect on the heart, because the heart needs the epinephrine to beat faster, whereas the liver, in response to epinephrine, starts to metabolize glycogen, release glycogen for metabolism, so that you're putting more sugar into the blood. But because that same hormone is received by 2 different cells, the 2 different cells have a different response to it, just by virtue of the fact that they're different cells. All right, as with everything in the body, there is always the potential for a problem. So if something happens to the signal, or you have a protein, for example, that is misshapen and does not phosphorylate properly, then you can have problems with signal transduction. It makes sense, right? There's lots of steps, and if at any step in the process of signal transduction something gets blocked, something doesn't work properly or whatever, then you don't get a signal being transmitted. And again, you looked at this in the Pogles as well, plus you had a little video that you looked at online where you were looking at how signal transduction pathways relate to endometriosis. So again, when you have problems with a signal, you can get diseases, right? Diabetes, heart disease, autoimmune diseases, all of these are linked to poor signal transduction pathways in some way. And again, sometimes that's genetic. The gene that's supposed to encode a particular protein kinase, for example, has a mutation in it, and the protein kinase is misshapen and doesn't work, right? So you can see how that can kind of happen. Also, though, drugs, toxins, poisons, all of those things that you see listed here can cause problems by either binding or blocking protein kinases from working. And sometimes these can be permanent, like if you have a toxin that permanently can do that, a poison or a venom or anything like that can sometimes cause permanent blockage of signals. And then you have problems with being able to receive a signal or having a cell that's insensitive to a particular signal because it's not reaching the end of that signal transduction pathway where the cell is able to respond. You also can have temporary things like anesthetics and antihistamines that can temporarily block signals. And for various reasons, like an antihistamine is trying to stop the cells from producing histamine, and histamine is what makes you all snotty when it's allergy season, right? So that release of excess mucus is because of histamines that get produced by cells in excess in response to a particular antigen in your body, like pollen or whatever. And therefore, if you take something which blocks the production of histamine or an antihistamine, so it blocks the signal transduction pathway that produces histamines, then we can block those side effects that we get from allergy season and make ourselves a little bit more comfortable. So we can actually kind of use that, our knowledge of signal transduction, to kind of make life better. As you can see, though, this is a lot easier than what we've been doing before. And as always, feel free to pop in and ask questions anytime you need. But that's how signal transduction works.